In this lecture, we will explore DNA repair, an essential process for maintaining genome integrity. We will cover various types of DNA damage, repair pathways, their roles in cancer prevention and aging, and emerging research topics in this field. Let's begin exploring the fascinating world of DNA repair. Narrator: to comprehend DNA repair pathways, it's vital to grasp the origins of DNA damage. Damage can stem from internal factors like replication errors and reactive oxygen species, or external factors like UV radiation and certain drugs. These insults generate various DNA lesions. Narrator: cells have evolved diverse DNA repair pathways to combat the various types of DNA damage. These pathways can be broadly classified into direct reversal, single-strand break repair, and double-strand break repair mechanisms. Direct reversal repair involves the direct reversal of specific DNA lesions, while single-strand break repair primarily deals with damages affecting one DNA strand. On the other hand, double-strand break repair mechanisms are responsible for repairing severe DNA damages affecting both strands. Narrator: base excision repair, commonly abbreviated as BER, is a highly conserved pathway involved in the repair of small, non-bulky DNA lesions. BER primarily deals with DNA damages such as oxidation, deamination, and alkylation. The process involves the recognition and excision of damaged bases, followed by the subsequent repair synthesis and ligation. BER plays a crucial role in maintaining genomic stability and preventing the accumulation of mutations associated with various diseases. In the nucleotide excision repair, NER, pathway, DNA damages caused by UV radiation, chemicals, and carcinogens are recognized and removed through a coordinated process. This repair mechanism is crucial for preventing skin cancers and is affected in xeroderma pigmentosum. Narrator: mismatch repair, MMR, is a crucial pathway that identifies and rectifies errors during DNA replication. It recognizes and eliminates incorrectly paired nucleotides, restoring the accuracy of the genetic material. Defects in MMR lead to hereditary non-polyposis colorectal cancer, HNPCC, underlining its importance in preserving genome stability. Narrator: double-strand break repair, DSBR, is a crucial process that fixes severe DNA damages caused by ionizing radiation, oxidative stress, or replication errors. Cells utilize two main pathways, non-homologous end joining, NHEJ, and homologous recombination, HR, for repairing double-strand breaks. NJ is fast but prone to errors, whereas HR uses matching DNA sequences for precise repair. These pathways are essential for preserving genomic stability, preventing cancer, and avoiding genetic disorders. In this video, we will explore the DNA damage checkpoint pathways. These pathways play a crucial role in preventing cell cycle progression when DNA damages are present. They ensure that damaged DNA is not passed on to daughter cells. Essential to these responses are protein kinases that induce cell cycle arrest, facilitate DNA repair, or initiate cell death if the damages are severe. Narrator: successful DNA repair relies on an array of proteins that coordinate and execute the repair processes. These proteins include repair enzymes, DNA helicases, nucleases, polymerases, and many others. Each DNA repair pathway has specific proteins that act as key players in the recognition, excision, resynthesis, and ligation steps. The dynamic interplay between these proteins orchestrates the efficient and accurate repair of various DNA damages. In this section, we will discuss the importance of DNA repair in preventing the formation of cancer-causing mutations. Mutations in DNA repair genes can lead to genomic instability and a higher risk of cancer. For example, errors in DNA mismatch repair can cause microsatellite instability, which is seen in certain hereditary and sporadic cancers. Other repair pathways, like NER and DSBR, are also involved in cancer development. Studying DNA repair's role in cancer is crucial for developing personalized treatment approaches and targeted therapies. Narrator: besides the general DNA repair pathways, specific mechanisms have evolved to repair unique DNA lesions. These mechanisms include repair of oxidative DNA damage, repair of DNA interstrand crosslinks, repair of DNA adducts caused by environmental toxins, and repair of DNA damage induced by chemotherapeutic agents. Understanding these specific repair mechanisms enhances our knowledge of how cells handle diverse DNA damages and helps in the development of targeted therapeutic approaches. In this video, we'll explore how DNA repair mechanisms become increasingly important as we age, as our cells face more DNA damage. Understanding this interplay can offer valuable insights for healthy aging and managing age-related diseases. In this video, we will explore the fascinating field of DNA repair research, focusing on exciting emerging topics. We will discuss alternative repair pathways, the connection between DNA repair and cellular processes like transcription and chromatin remodeling, the impact of DNA repair on immune responses, and novel therapeutic strategies targeting DNA repair processes. Understanding these topics will enhance our comprehension of DNA repair's complexities and its relevance in different biological scenarios. Summarize in 23 words. DNA repair is essential for maintaining genomic stability and preventing mutations. Understanding it is crucial for personalized medicine and developing innovative treatments for diseases like cancer and aging.